Okay, everyone, I'm going to go ahead and get started because I've got a lot of stuff to share with you today, and I don't want to have to rush through it any more than I already do to make sure that I cover all of the bases. But thank you all for coming today. So those of you that are joining us on Zoom, and many of you who are in the room here, this is really great. My name is Amy Johnson. I am your technical specialist for spatial biology platforms from Nanostring. So it's very nice to meet you all. And what I'm going to do today is go over our two spatial platforms that are available. Some of them you may have heard of, some of them you may not, just because they're new. So we're going to talk about the GeoMx Digital Spatial Profiler system, and then we're also going to be talking about the CosMx Spatial Molecular Imager platform. Okay, so why are you here today, or why are you logging on to the Zoom? I'm going to bet that some of you have, in papers that you read, posters you've seen, grants you've reviewed, you've started seeing terms like spatial transcriptomics, spatial proteomics popping up in these different arenas. Maybe, just talking with colleagues, you hear that these are technologies that people are using more and more. So what I've seen is over the last year and a half, a real explosion in spatial biology platforms. The types of applications that are being used, the research focus areas, and so on and so forth. So it's really a very, very exciting time to be interested in spatial biology, for sure. Why am I here? As I said, I am your technical specialist for these platforms specifically. So a lot of what I do is coming to events like this, giving you an overview of the platforms, maybe some ideas of how you can use these platforms in your research. And then I'm here to talk through if you have ideas about study designs or feasibilities, things like that that you're kicking around. Feel free to contact me for any of that. I'm also joined today by my colleague, Nicole Golden, in the back in the red shirt. So she is your consumable sales specialist or representative. So if you want to run projects, she can also take you through study design and get you costs and things like that for how to do these types of assays. So I mentioned at Nanostring, we have two spatial biology platforms, but I'm going to back up for just a second because Nanostring's been around a lot longer, predating these spatial biology platforms. So some of you may be familiar with the nCounter system. So this is a system that is designed to do kind of mid-range plex, hundreds of genes, bulk gene expression analysis. The nCounter is a true workhorse. Many up and running, continuously used in the field today. So I just wanted to mention and make sure that you were aware of that system today, although we're not really going to be spending a lot of time on it. We're going to talk about those two platforms, the GeoMx Digital Spatial Profiler and the CosMx Spatial Molecular Imager. So you might be wondering, why do we have two platforms at all? Why can't you just design one system that will cover everything? And the fact of the matter is that biology is wide-ranging. People's research questions are wide-ranging. And so to be able to provide information and tools for researchers on the whole spectrum between, pure discovery, you've got a new model system, you've got a new knockout mouse or a new cell line that you want to phenotype and you have no idea what to expect. Pure discovery type work. The GeoMx system you're going to see is best suited for that. If your research focus is really at, I need to see what cell A is doing next to cell B and how they're interacting with each other, true single cell and subcellular resolution, the CosMx Spatial Molecular Imager is going to be the best avenue for you. So there's a huge breadth of biology. There's so many scales, and that really, in order to tailor things to cover as many researchers as possible, we have these two platforms. We're going to start off with the GeoMx Digital Spatial Profiler. So this is the system that was launched in 2019. And this is the system that's able to do very high-plex transcriptomics and proteomics. So what you see on this slide here, let me go here for my Zoom people too. This is just a slice from kidney. Okay, so kidney, pretty complex organ, very highly organized. But the area where you find different structures and cell types dictates the function of those different areas. So with the GeoMx, you can take a very complex organ like this, and you can look at specific cell populations within this organ or functional units, different ducts, things like that. And so you can design your study that you can look at gene transcription differences between different cell populations or different areas within the tissue. We see a lot of people using the Geomx to address questions around tumor heterogeneity, if cancer research is your thing. You know, one area of the tumor looks a certain way. What does the area next to it look like in terms of gene and protein expression levels? Tumor versus tumor microenvironment. Host versus, you know, so in infectious disease, if you're looking for an area of infection, what does the microenvironment around that site of infection look like? And those types of questions there. So very high-plex, it's truly a profiler. We can do both protein assays. Oh, sure, go ahead. I don't think I can drag it, but I might be able to hide it. There we go. How's that? Better? Okay, thank you. Okay, so protein assays, if those of you that are used to doing immunoassays, multiplex type assays, you know, a lot of times you can do about 30 to 40 plex, unless we're talking about fluorescence. But the way that the Geomx works, if you threw everything that we had at it off the shelf, fully validated, ready to go, you would be running 150 plex protein assay in space in different cell populations, depending on how you design your experiment. If we're talking about RNA assays, so gene expression levels, a year and a half ago, we launched 2 whole transcriptome atlasing assays, one for human, one for mouse. So if we're doing a human whole transcriptome atlasing assay, you're looking at the expression of a little over 18,000 genes simultaneously in space. If we're talking about a mouse whole transcriptome, it's a little bit more than 22,000. So these are all the protein coding genes in the genome. The platform is pretty scalable. So you'll see with both the GeoMx and the CosMx, the instruments can hold 4 slides. And depending on how you design your study, you may be able to put multiple samples on each slide. So now you can scale up throughput there and the amount of data that you're generating. Everything that Nanostring develops and designs is really designed with the intention to make it as flexible as possible. So we see this in terms of the types of cells or the types of samples that you can work with. Those of you that are familiar with Nanostring, especially if you've used the Encounter, you know that Nanostring has built their reputation on working with FFPE samples for gene transcription. So samples that typically you see very low RNA quality scores in just because of the process of embedding itself. These also tend to be the blocks that are archived in people's labs. I know that was certainly the case when I was in grad school and doing my postdoc. Drawers full of FFPE blocks from different studies that had been done. So we know with both of these platforms that you can take that archived tissue now, even decades old blocks, get some fresh sections and do these types of assays on them. So think about stuff that you already have collected and sitting in the lab and whether or not this would be an appropriate analysis to do. So FFPE is all good. A really difficult sample type to work with when we're talking about transcriptomics. So we start there. We optimize and validate all the assays there, because if it works there, it's going to work with everything else. Fresh frozen tissues, fixed frozen tissues, so you have a lot of flexibility there. We do also have the ability to customize assays. So again, if we're talking about RNA expression, say you're running a whole transcriptome assay, but there are a couple target genes that you want to add in. Long non-coding RNAs, maybe viral genes, you know, depending on the type of work that you're doing. We can make those probes and you can spike them in to that whole transcriptome assay. You could also build a totally de novo from scratch custom assay there. Same goes for protein. So we have modules, we have assay kits already validated and ready to go. And then 3 different ways for you to add in custom protein targets there. One of which is a new kit that we launched where you're able to do the barcoding necessary on the antibody in your lab and just add it into your assays there. So that's pretty exciting too. So the Geomx technology, how it works is really based on the reagents that we're using for these assays. So the next couple slides, we're kind of going to go over the workflow of a typical Geomx assay. And you're going to see that we're going to stain your samples with two different types of reagents. And what I'm showing you now on this slide are the profiling reagents. These are the reagents that are used to actually quantitate the expression levels in your tissues. So if we're doing a protein assay, the affinity reagent is a primary antibody against your protein of interest with a DNA barcode attached to it, and it's attached via a UV light cleavable linker. So you'll see why that's important in just a second. If we're doing an RNA assay, it's an in situ hybridization probe, still with a DNA barcode attached to it, still using a UV light cleavable linker there. So let's say you have an FFPE block, you've taken a section from that block, typical 5 micron section. You put it on a standard positively charged microscope slide. Again, stuff that you probably already have in your labs right now. And then we are going to hybridize those profiling reagents onto your tissue, depending on the type of assay you're running. And we're also going to stain your tissues with what we call morphology markers. So the GeoMx Digital Spatial Profiler is a 4-channel fluorescent microscope. We save one of those channels for a DNA stain, and we need that so that we can focus the microscope. So that leaves us 3 channels now that we can use reagents, tagging specific cell populations or specific structures in your tissue, so we can visualize the tissue. We're not doing any quantitation with these. It's purely just to, so we can get an image of your tissue there. So these are typically antibodies, fluorescently tagged antibodies. We do work with labs that use RNA scope probes as morphology markers as well. Again, mostly it's sort of an infectious disease type setting there. Okay, so your sections are on the slide. We've already prepped them by staining with the morphology markers, and we've already hybridized with those profiling reagents. So now we're ready to put the slide on the instrument. Let me click through this a little bit. So the first thing that's going to happen in the Geomx is that it's going to take a scan of your slide. So we can stitch together the fluorescent image of your tissue, what you're looking at there. You need that information because you're going to tell the instrument what areas within that tissue you want to focus on to profile more deeply. You can put these, we call them regions of interest or ROIs. You can put them wherever you want. The limit on how many is extremely high, pushing close to 800 on each. And then there are different strategies you can use within those areas to make sure that you're getting the data that you want. So you go through all of that and you put down your regions of interest and you set your selection strategy. So then what happens next is you hit go, you hit run on the instrument. And what the Geomx is going to do is it's going to go to each of those ROIs and it's going to shine UV light just on those areas. You're going to see that we can focus that light so much that we can look at specific cell populations within those areas. So shining the UV light on those areas is going to lead to the cleavage of those DNA barcodes off of the profiling reagents. Those barcodes are immediately aspirated. They're sucked up using a microcapillary and then placed in a 96-well collection plate. And all of this happens automatically on the instrument. You're not sitting there with like a P2 trying to suck up these barcodes. And this process just repeats and repeats until the barcodes from all of the areas you've designated have been collected. So now we need to count those barcodes because that's our raw data, that's our expression data. If you're doing a mid-plex assay, which a lot of the protein assays are, you can count those barcodes on a Nanostring encounter system. Works really well. If you're doing a whole transcriptome atlasing assay, we're going to count those barcodes on an Illumina next generation sequencer. But I want to point out here that we're not sequencing native RNAs. We're sequencing very short synthetic DNA barcodes. So the read depth or your burden of sequencing is nothing like it would be if you were doing a from scratch NGS type of experiment. A circle that's 10 microns in diameter. So for those of you on Zoom, the question was how small can an ROI be? And it's a circle that is 10 microns. So then we have all that count data. Let's say we got it from the Illumina sequencer. we have that data file, there's a conversion that needs to happen, and then we can put that file into our data analysis suite that comes standard on every GeoMx instrument. So this suite is really designed to allow you to, first of all, link the imaging data you have, so the IF image of your tissue, with the expression data that you've now collected. And so you can go and say, okay, in this area of my tissue, I want to see what the differential gene expression is right here. And then if you click that in the software, you will see the related data show up. You can do pathway analyses, you can do hierarchical clustering and make heat maps, differential gene expression, dimension reduction, and so on and so forth. So we've built, pretty standard bioinformatics and statistics tools into the data analysis software suite. Really designed for people to do bioinformatics who are not bioinformaticians, like me. If you do collaborate with a bioinformatician or another group, that's totally fine. You don't have to use our software. You can take your raw data and do whatever you want with it. We see that happen a lot as well.
So in between step 3 and 4, if you have more regions of interest, like more than 96,
yep.
How does the machine support a second plate?
It'll prompt you to put another plate in.
Okay. And do you see any variability between plate 1 and plate 2 at that time?
No. And you can have up to 8 of those 96 flow plates in a single run. Any other questions? Yeah.
It's about quantification. So you're trying to make inferences, relative levels of expression. It sounds like it's directly linked to the efficiency of the barcode and quantifying.
Yep.
Can you tell us anything about that?
Yeah, I mean, to the point where there is a normalization process that you would have to put your data through to make sure that the counts, which is your expression data, it's real data and it's not background, or something else didn't happen there, let's say. But then it's really, in terms of, are you asking how you would express the data, or?
I'm asking if you're looking, let's say you're comparing 2 ROIs.
Yep.
On this platform. If you really wanted to, if you were doing sequencing, you could just quantify transcript.
Right.
But we're actually quantifying barcode.
Yes.
So I just was wondering, it's sort of an indirect way of quantification because it's related to the efficiency of barcoding.
Well, there's one barcode on one probe that correlates to a messenger RNA from a single gene there. So it's still that direct. You're only going to get one of those probes binding to one mRNA there. So it's more direct than, say, something like a Western blot in that case there. But when you are doing those barcode collections within a single ROI, let's say you've designed your strategy so you want all the barcodes from all the T cells in a specific area. So you can do that. There's definitely strategies. We see it all the time. The count data that you're going to get from the T cells in that ROI is going to be an aggregation of all the T cells in that ROI. So if you have 50 T cells in there, it's all the expression data from those 50 T cells. So that's, for Geomex, definitely think of this as looking at single cell populations. Single cell. Yeah, so we can, I'm happy to get an R&D person on because it goes through all of that validation process in the development there. There are guide rails on how thick your sections can be for that exact reason. If the UV light can't penetrate your sample well, you're not going to get efficient cleavage. But that's why we've already worked out all of these conditions and have tested all of this there.
Does that mean, actually, you asked the question much better than I did. Ultimately, I would really like to know if you compared the quantification of the barcodes with, say, direct sequencing cells from that region.
Let me make a note of that because I want to say that's been published already. So, or actually, Nicole, can you just make a quick note of that for me? Thanks. Okay, so I hinted at this briefly. Looking at the study design, the assay study design, this is specifically looking at the whole transcriptome atlasing assays. So as I said, we are working with pools of in situ hybridization probes against, let's say, 18,000 genes. We're doing a human assay. That the assay is designed specifically so that you are going to pick up, a very wide dynamic range of expression levels. You're not just going to get the high expressors. Okay, like can happen in other technologies, you're going to get the medium and the low expression levels as well. And that's because we're actively looking for these mRNAs within your tissue. We're not just pulling things down. We can show that you get that wide range in specific cell populations because we can focus that UV light to follow the contours of the cell populations within your tissue. So let me put this another way. Let's go back to the tumor example. Say we have a section of tumor that we've used pancytic keratin as a morphology marker. Just going to tag the epithelial cells that are there. Say we've also used CD68 for the macrophages to tag those there. And then say we've used CD3 for T cells. Okay. So now within a certain circle or polygon or rectangle, whatever shape I've drawn in that area, I can tell the instrument first go to this area, shine that UV light only on the pan-CK positive cells. What's going to happen then is that there are millions of micromirrors in the Geomx that are going to configure themselves to follow the edge of that fluorescent staining for the pan-CK positive channel. And that is going to shape the UV light. Follow that shape so that you're just shining UV light there. So we put the UV light on, we've collected those barcodes, but you've set up your strategy that now you want to know what are the expression profiles in the macrophages. So the instrument will go back to that same area, reconfigure those mirrors, and shine the UV light on only the CD68 positive cells. You also are interested in T cells. You can go back to the same area again and say, now give me just the barcodes from the T cells. And lastly, go back one more time and give me the barcodes from everything else that's left, anything that has a nucleus there. So now within this one area of the tissue, you've compartmentalized things into specific cell populations. And so you can look at the expression profiles of macrophages versus the epithelial tumor cells versus the T cells, all without destroying the sample at all there. So this is segmentation. And to be honest, this is the strategy that we see most people using for their Geomex assays at this point, rather than just drawing a circle or a square and taking the barcodes from everything that's in there. Mm-hmm.
So you draw a circle or whatever, and then you tell it, I want it to focus on this type of structure in this circle. So the shape doesn't, it only outlines the border. It's really about what's within that.
Yeah, based on the fluorescence.
Okay.
Mm-hmm. Yep, exactly right.
So could you theoretically, I know you said the 10 micron was the smallest limit. How big?
The largest you can draw is a rectangle that's 660 microns by 785 microns.
So could I theoretically just have one big ROI and be like, I just want you to focus on this specific subset?
Yep. Absolutely. Just a really quick example in the interest of time, kind of demonstrating that segmentation strategy. So this is a colorectal cancer sample. You see the morphology markers used listed here below. These circles are the different regions of interest that have been selected, some within the tumor, some at the margins, some in normal adjacent tissue. And now this is actually from a protein assay. Because we've compartmentalized and ordered those barcode collections, we can now ask questions not only about, tumor versus normal adjacent, but now specific cell types within those areas. So where they are, stroma versus tumor, and you can start teasing things apart this way. Up until about, I want to say 6 months ago, if somebody wanted to do protein and RNA assays, a recommendation would be to take serial sections from your block, run your RNA assay on one, run your protein assay on the other. We've since worked out the conditions that you can do both simultaneously now on a single slide. So spatial proteogenomics, if you're really interested, you can scan this. I'll bring the slide back up afterwards, and you can get the protocol but we've worked out all the conditions, tested all the iterations of the protocol to show that this is robust. One last thing before we switch gears into CosMx is that there are people that have a lot of whole transcriptome data. They also have single-cell RNA-seq data, maybe from the same samples, maybe they have access to a data matrix from somewhere. The Broad posts a lot of matrices in a free database. What we could do is spatially deconvolve that single-cell RNA-seq data. You run a whole transcriptome assay on your sample. We have an algorithm built into our data analysis suite that will look at your whole transcriptome data, look at that single-cell RNA-seq matrix that you have, look for commonalities between the two, and then show you enrichment areas for different cell types within your tissue. So spatially resolving that single-cell RNA-seq data set there. So we see this used a lot as well. So a nice way to really bridge together two pretty powerful technologies. Okay, speed to publication. I spent over 15 years in academic science in one capacity or another. Of course, publish or perish, right? We hear that all of the time. With people using the Geomx, we've seen kind of unprecedented timelines from the time of getting your data to when it's published. Things that may have taken me years to publish when I was in grad school or doing a postdoc are taking some people months to publish in really high impact journals. So we're approaching 150 peer-reviewed publications with Geomx. The average impact factor is 17. We see these data sets coming out in Nature Communications, Cell Reports, like I said, pretty high impact journals. Okay. So for those of you, has anybody in here run a Geomx assay already with the molecular pathology core? Okay. For those of you that are thinking about doing that, or maybe already are sitting on some data that you need some help analyzing, we do have a spatial data analysis service where our bioinformaticians will take your data set and do the comparisons that you've outlined for your project. So we're running a promotion on that service there. If you need more details, you can definitely ask Nicole in the back and she can tell you what that process looks like. Okay, I am going to switch gears to CosMx, but before I do, are there other Geomx questions? Oh, okay. Per slide. So it's 896 well plates is for a run. So what is that? 768 collections. Yes, you can. So we do have people that use maybe an approach more akin to kind of flow cytometry for picking out different immune populations, where you can, you use your morphology markers, but you can segment on, I'm trying to think of a quick example. You all could probably come up with something better. But a subset of T cells that are, high for one marker and low for another versus a subset that are high for both. Those types of, gating, for lack of a better word, approaches are possible as well. Is that it? Okay. Thanks, Nicole. Okay. So Geomx is a profiler capable of very high-plex assays. You're looking at single-cell populations. CosMx is an imaging-based system that's going to allow you to do truly single-cell and subcellular spatial transcriptomics and proteomics. The way that this platform works in terms of the samples that you can work with, your sample prep, the slides you're going to use, all the same as Geomex. So applicability FFPE, yes, of course, and so on and so forth. What's different about this system is that we are not collecting barcodes and counting them somewhere else. We are still working with reagents that have barcodes attached to them, but we're reading out that barcode in situ within the tissue. And we're collecting that data by imaging. So if you look at this slide, I believe this was from a melanoma project that was run. And you see within a specific field of view, so a scan area of the instrument, you have the tissue and you see all of these teal lines. These are the cell membranes. So what you're going to see with single cell spatial platforms is that cell segmentation is absolutely critical. We'll talk about this more because we have to figure out exactly where one cell ends and the next one begins. Through the course of these assays, whether we're doing RNA or protein, we are going to find the exact XY coordinate of every messenger RNA that's scanned or every protein. And in order for that data to be useful to you at all, it has to be assigned to a specific cell or it's kind of garbage DNA or it's garbage data. So we spent a lot of time working on the cell segmentation strategy that we use. But this is a slide that our CSO shows in all his talks, just to show you the amount of data that you're able to collect in a single CosMx run, which a single run will give you about 15% of the data collected in something like the Human Cell Atlas. So for those of you that are really focused on cell atlasing, cell type mapping in tissues, I think that you will find that pretty impressive. Okay, so this is kind of a look at what we're getting at with CosMx. This is lymph node tissue. We've scanned across the whole tissue here. And then what we're doing, let's say we're doing this RNA assay. The assay that we're running now is a 1000-plex human universal assay. Not a whole transcriptome, 1000-plex. So the trade-off for the resolution is the plex size of the assay that you're going to run. And that's the case regardless of any imaging platform that you're working with. Not a biology platform or problem. It's not a chemistry problem. We've shown we can build the assays. It's a physics problem. And we are trying to tell one fluorescent spot from another fluorescent spot that's very close to it. And it's called optical crowding that we're up against there. So let's go back to this example. We're running this 1000-plex RNA assay. The way that this assay is built is that we are going to be able to do cell typing and cell atlasing. So of that 1000, about 250 of the probes are specifically in there to identify different cell populations in the tissue. The rest of the probes are there to tell us about cell function and cell state and receptor ligand interactions and types of information like that. But what you're showing here within a specific field of view, these colored dots in the middle, those are different cell types. Those are cells that have been identified and given some sort of identity. But this is what the real data looks like. And so now you can start to see where there's subcellular localization comes into play too. Each of the dots in the circle all the way on the right-hand side are an individual messenger RNA there. So if we go back to, thinking about things in terms of single cell, because this is really a direct corollary to single cell RNA-seq type data, where if you're doing a single cell RNA-seq assay and your data in the end, a lot of people look at it in terms of TISME plots and UMaps, you figure out what are the cell populations that were there in your tissue, right? Your parts list of your tissue. But you don't have any information about how all those cells were arranged in space and how they're interacting with each other, because you've had to dissociate everything to do the single cell run. So what we're doing with CosMx is we're getting you that data, what cells are there, but you're also getting all of that spatial information. Where were all of those parts within the tissue? The chemistry for these assays is slightly different than with GeoMx, but as I said before, we're still working with in situ hybridization probes and primary antibodies with DNA barcodes attached to them. Instead of cleaving off those barcodes, we are instead flowing over pools of reporter reagents, fluorescent reporter reagents. With each pool, if a reporter finds its complementary sequence, it hybridizes, everything else is washed out, an image is taken, everything is cleaved, the next round goes in, and that process repeats. So these assays are pretty data intensive because we're going to end up with all of these stacks of images at the end of a given run. We have projects that we've already run with partners that you've seen here with various tissues. And so I'm going to show you just kind of a couple examples to kind of let you know how other researchers are thinking about using these platforms already. The first of which is back in October, Nanostring released a data set from CosMx on non-small cell lung cancer. So this is from humans. These were FFPE samples, but just to demonstrate the number of transcripts that we're able to detect how many cells we're scanning in any given run. And this data set is on our website. So if anybody in here likes bioinformatics, feel free to download it and play around with it as well. It's also the data set that was included in the technology article describing this platform. I put this up here because this kind of drilled in for me. When we say that we can work with samples that have very low RNA quality, we really, really mean it. And so from those same lung samples, if you look at the DV200s for lung 5 and lung 13, which are 20%, you know, very, very low RIN scores, although we could debate, you know, which is a better measurement, but we're still able to get 97% of the cells within those samples gave us data that passed our QC qualifications there. So really, truly, degraded samples are okay on these platforms. So let's say we've done a CosMx RNA run. The first step is going to be doing that cell atlasing, figuring out what's there and where is it. But then using that same data set, because we have the XY coordinates for all these mRNAs or all these proteins, we can start doing more sophisticated analysis. We have tools that we've built to identify different niches or neighborhoods within tissues based on expression profiles, areas that might be functionally related to each other. In that 1000-plex assay, we have probes against 100 canonical receptor ligand assays or receptor ligand pairs. And so now, because again, we're truly looking at single cell, we can look at a specific cell and we say, well, you know, this cell's really increased its expression of receptor whatever. Now you can look at the cells around it and has it increased its, have they increased their expression levels of the cognate ligand there? And so, you know, again, information that previously I can only think of maybe one or two other ways that you could even approach this, and they weren't even really great methods there. So now, you know, very specifically drilling down. And then of course, differential gene expression profiles. How does one area, one cell, et cetera, differ from another there? This is kind of an example of that receptor ligand interaction data set. So this is going back to lymph node, different zones within the lymph node. But basically the take home message that I want you to take home from this slide is that we can identify different cell types. So with the CosMx assay run, we show that the CD86 B cells from the light zone co-localized with CD28 cells there. So now we know like who's next to each other, what's happening there. But then who's talking to who? You know, which cell type is influencing the phenotype or the activity of the other cell type. And so now if we did a GeoMx run on a serial section from this, we could start doing higher plex pathway analysis. And you can say, okay, well, let's look specifically at the CD28 pathway. And is that being regulated? And so you can kind of start to get an idea of how these platforms kind of feed into each other there and are related to each other. Mouse brain. So we talk a lot to people doing neurobiology applications. So the brain being an extremely highly organized organ where, you know, where things are dictates the function of that area and so on and so forth. So it's very important for us to develop these assays. This is just data from cell atlasing from mouse brain. This is actually from a lab at Wake Forest that I work with, and really just confirming that the different cell types that we are picking up do actually line up with, you know, canonical neurobiology about mouse brain anyway. So kind of a validation experiment there. If we drill down on this data, we start to see kind of the power that we can derive from this. So in a mouse brain, in a specific area of the mouse brain here, let's say we've identified these different cell types: radial glia cells, neuroblasts, granular neurons, and astrocytes within a specific field of view. Okay, so that's just going to click through this, which is great. So we found a bunch of microglia, let's say, but is a microglia a microglia? So depending on where that microglia is, what type of cell it's interacting with, you know, what can we find out about the function of this specific microglia in this part of the brain here. So we have the probes that tell us where the microglia are, but then we have all these other probes that tell us about the function of the microglia. So we start looking at expression levels of phagocytic markers in these microglia. The microglia are macrophages. They're a type of macrophage. You know, they're there to eat things. And so having a marker of phagocytosis is really, really critical. So this is what's shown here. So here, an interneuron and a microglia, and we see, you know, some low expression, these blue dots of maybe some phagocytic markers there. But what if we looked at a slightly different part of that area of the brain, of the dentate gyrus, and we look at different microglia that are associated with different cells, and then we start to see some pretty critical differences, where this is the first microglia we looked at. These two microglia that are associated with different cell types are showing different functional patterns. When we look at these two microglia that are associated with these interneurons, and they are very phagocytic. They're expressing high levels of phagocytosis markers there. So maybe this is engulfing a damaged cell. Maybe this is, you know, pruning of processes and synapses and so on and so forth. But this is the type of information that you're going to get from a CosMx run. You're also going to get, you know, fluorescent data, just like with Geomx there. So you can kind of, you know, look at your samples in a couple different ways. Okay. We have a study that we performed with Grant Kohler at St. Louis University that's near and dear to my heart just because I'm a biochemist. And so I spend a lot of time thinking about fat and liver and the pancreas and insulin and all that stuff. So I was very excited to see this data set come out. So you can see this is just overall statistics of this study. And the goal was to look at islets from 3 different patient populations: people that were metabolically normal, people that were type 2 diabetic, and then a population of people that were obese but metabolically healthy there. So is there anything different about the islets within these different populations? So if we start with the CosMx run, and Grant was specifically interested in looking at the beta cells, the alpha cells, and the delta cells. So the cells that are producing the regulatory hormones in the pancreas. And so we just look at his cell atlasing data. You can see that there are pretty huge differences in terms of the types of cells that are found in the islets from these different patient populations. Where in normal people, almost 90% of their islets consist of endocrine cells, like you would expect. But that shifts dramatically when you look at the type 2 diabetic population and then the metabolically healthy obese population. So a population that is technically not diabetic, but their islets look very different than a normal patient there in terms of the types of cells that you find there. So, okay, we've got this information that these islets are really different in terms of cell composition. But even among those groups, if you find beta cells in the islets from those 3 populations, is the beta cell from a normal person showing the same expression profiles as a beta cell from a metabolically healthy obese? So this is where, feeding off of that CosMx data, we go back to Geomx. And from the same area in that pancreas, we can now do a whole transcriptome atlasing assay specifically on those cells and look at the pathways and the characteristics of those cells there. So giving you a much more complete story. And then lastly, I'm going to round out with some protein. We spend a lot of time talking about RNA assays, but just to remind you, both the Geomx and the CosMx can run protein assays as well. We described a 104-plex protein assay on CosMx at AACR. If this is of interest to anybody, I'm happy to send you the poster and we can go through it more detail. But just to show you that this is the plex size that we are achieving with this platform. So very quickly, I'm not going to spend a ton of time on this, but we did a study using core needle biopsies for breast cancer patients. And so this is an instance where, you know, using, you know, different identification strategies to identify the immune cells within the sample here. So we have this protein data, and we can see there are tumor-associated macrophages here. If we look at the correlative RNA data, we see that the markers being expressed for the tumor macrophages are co-localizing where the protein markers for tumor-associated macrophages are. There's also kind of, we found this population of macrophages kind of up here within the tumor, so it has actually infiltrated the tumor. Anybody involved in immunotherapy type design knows that you want to target the immune cells that are infiltrating tumors or understand why they're infiltrating, because that bodes well for therapeutic response. And what we found is that in that population of cells that we've identified is that they are expressing specific genes different from the other tumor-associated macrophages. So this is all within the same biopsy there, putting together protein and RNA data. Just to give you an idea of the types of assays that are available and in development for CosMx, as I mentioned, the human universal cell characterization, that's our 1000-plex human assay. We've put a lot of attention to building the neurobiology assays because they've been in most demand. So we're starting with mouse. Human is very closely behind. And then that'll just continue to build out and build out. As with Geomx, we have the ability to customize, whether this is adding in custom probes or antibodies to an assay or totally starting from scratch there based on what you need. So we have the samples, we have the CosMx itself, but then what are you going to do with all this data? I told you this was data intensive. This is terabytes of data you're generating with everyone, all of this imaging data. In order to not only handle that data, store that data, process it, be able to analyze it, we've built a cloud-based spatial portal. It's called Atomics. So what's going to happen with CosMx is during your run, as data is being collected, it's going to be passed off to the cloud almost immediately there throughout the run. We have to process those samples to flatten them so we can segment. And then from there, all the different types of analyses that you want to run. And so again, this will be cloud-based. The huge advantages are it's going to allow you to collaborate with people much more easily. You're not going to be emailing terabyte file sizes to a collaborator to do data analysis. You both can be in there at the same time working together. So ease of that, but also processing speed. Just looking at cell segmentation, if we did a run where we did the cell segmentation on in-house servers at Nanostring in Seattle, compared them to the cloud-based portal that we've built, the cloud is 75 times faster. It can do in an hour, which took 75 hours to do on our in-house server. So I think, just being able to leverage the computing power of the cloud is going to be really critical for anyone doing these runs. So we've developed this because of the data that's coming off the CosMx. You will also be able to integrate your GeoMx data there as well. So really creating a whole ecosystem for scalable storage and analysis, collaboration, getting you to publish and present data even faster. Just the types of analyses that you'll be able to do right off the bat in the cloud, things that we've covered. So of course, doing dimension reduction, ligand receptor interactions, nearest neighbors, kind of that niche and neighborhood identification. These all come standard. Just like with Geomx, if you want to take your data and do something else with it, totally fine. Very flexible. We're very mindful of making sure that all of the files that we're using are compatible with open source and third-party software packages. Basically, it comes down to that the field of spatial is moving so fast, and people are continuously designing new tools that we want to make sure that you can take this data and go wherever you need to with it. So I believe this is my last slide, just a final comparison between the two platforms. We started off talking about the GeoMx DSP, that it's capable of profiling unbiased, very high-plex assays for both RNA and protein. It's automated. You can run a whole host of sample types on it, and you will be able to access the Atomics cloud-based portal as well. CosMx, on the other hand, truly single-cell subcellular resolution, 1000-plex RNA assays, 104-plex protein assays, but you're able to answer questions that you wouldn't normally because of the resolution of the system here. So I'll end there. I'm happy to hang out and answer any questions that you have. A lot of what I've been doing lately is specifically with CosMx, just talking to people about their ideas and how they see this fitting into their research, which is fun for me because this is what we think people are going to start with, but we know that people, researchers are extremely creative, and so happy to see where they take these technologies. Yeah. Nicole, were there any other questions in the chat? Okay. Yeah.
I have two questions specific to the machine. So over for bio.
Is molecular. Okay. Yeah.
I don't know if it's the molecular.
Oh, it's
but-
different.
Yeah. No, all is good. So I noticed in your neuro slide on that experiment, they also have the gridding when you image the like your tissue. You also get that gridding. Is there an issue with the lack of, it looks like, anything fluorescent in those grids? I noticed you don't have any regions of interest on the grid.
So what you didn't see is that we're talking about CosMx.
No, Geomx.
Geomx, you
Yeah,
see gridding?
We have gridding. We took it, like I have had several meetings and no one can figure out why we're getting the grid. It looks like the machine stitches things together.
Okay.
And is there an issue if the region of interest were to be placed on that lack of fluorescence grid? Would you not get a read?
I want to say no, but the reason I'm hesitating is if you're not getting fluorescence there, I'm worried about your sample prep, which could influence the hybridization of the profiling. So that's why I'm kind of hesitant.
So it looks like it's a grid from the machine.
Really?
Yeah, it looks
Okay.
exactly like the image.
Okay.
Showed up there, there's a grid on it
From
the-
CosMx. I mean, we see that with CosMx.
Yeah. So
Um,
we see it in our Geomex machine
Okay.
over multiple experiments.
Okay. Who have you talked to at Nanostring?
There was
Have
a-
you talked to Nicole at all? Okay. Yeah, let's definitely get your information because we want to get that figured out.
And then I noticed here on the machine, our Geomex machine is not online. How hard is it to get the CosMx machine online? And y'all help with that type of support?
Yeah, so nuts and bolts. So the CosMx and the Geomx are both benchtop instruments. The CosMx is about this big right now. You plug it into the wall for power and you plug it into an Ethernet port for data. What we spend a lot of time doing, we meaning me, is talking to IT groups at different institutes because there are data security things that need to be worked through. And so I'm sort of the liaison between our IT people and the institution's IT people, just to square everything away, what's allowed, what's not allowed. So it's not unheard of. We're doing it all the time. Yeah.
Super secure. Love to take, sure all the very research stuff.
Yeah. And I mean, I will say that two of the institutes I just went through with this they're not academics. They are hospitals with clinical samples and patient information is the thing. And we were able to satisfy everything that they did. So.
Some sort of timeline to get online for those systems.
To get paperwork done, because that was a big part of it, I would say it probably took us a solid month back and forth if you put all the timing together. Yep.
So there's, I'm going to do a consortium analysis project.
Okay.
And there's a lot of, every month, there's back and forth discussion of uniformity forms. So we use that.
Yeah.
And I wondered if we could speak to two issues. The first is specifically to your platform, the limitation of the number of probes. Sounds like it's 1,000-plex.
For RNA, yeah, on CosMx, yeah, correct.
So design of those what you call probe sets, I
Yep.
suppose. How is that done? Is that what was noted on your slide about the pre-fab versions versus the custom versions? How customizable are your probe sets?
So even two things about that, we're sitting at 1,000-plex right now. It's the highest plex for a single cell platform on the market. So you'll see other platforms are around 300 to 400 genes. We're at 1,000 and actively working to increase that. And so, just know that that is already being worked on. For customizability for the 1,000-plex assay, you can add in up to 50 custom probes. If you wanted totally from scratch,
custom panel. The upper limit right now is 300.
So you have a panel of cell markers that's the stock version that you
Yes.
use.
Just off the shelf assay kit, and then you can add to that as necessary.
The other question is whether you could make any general comparisons and contrasts relative to the Visium system.
Mm-hmm. Yeah, so I would say Visium is the best comparison is to Geomex in terms of what the information you're getting from these different types of platforms. So for those of you that aren't familiar with Visium, the way that those assays work is that you work with slides that have typically 4 areas on them. Within each of those areas, there are circles that are printed with capture oligos, basically. So when you're doing the assay, you take your slice of the tissue, you put it in the area on one of those 4 areas. There are a series of prep steps that include permeabilizing your tissue. And so what's happening then is that the RNAs are being released from your tissue there. Those capture oligos are picking up the poly-A tail of those messenger RNAs. So a couple things with that, it's a pretty passive process. You're permeabilizing and hoping things flow out. Just because of the way that it works, you're going to be skewing your data toward the high expressing. It's just a numbers game. There's more of those mRNAs around, more of them are likely to bind. So, you have to think about your data in that way as well. The spots that are printed on the slide, they're 55 microns in diameter. They're 100 microns apart from each other. So if you think about that in terms of a tissue, which does not have spaces, right, between different cells, you're not going to cover an entire area of your tissue just because of the way that those spots are printed onto the slides there. The 55 micron slides, you're going to be picking up more than one slide, maybe a couple half cells, more than one cell, a couple half cells, that sort of thing. So all of this you need to think about when you're analyzing this data there. Where on the other hand, with Geomx, you can exactly tell the instrument what area you want to interrogate. We don't have to worry about those spacing issues. We also don't have to worry about biasing our data toward the high expressing genes because we are using probes to actively find these messenger RNAs there. Those are the 2 biggest things. I mean, to Visium's credit, you don't need an instrument to run it. Where with the Geomx, an instrument hardware is required there.
So far, this has been cost prohibitive for a lot of the projects in the consortium.
Really? Okay.
Where do you guys stand, not the numbers, but relative to this?
I don't know where their assay costs stand, to be perfectly honest, how much the reagents cost. We price things per slide. There's a certain area on that slide the instrument can see, so you can have multiple tissues on a slide. So keep that in mind as well. If you're doing a whole transcriptome atlasing assay, it is going to be, Nicole, help me out here. One slide, is it $2,500? Right, for one slide of a whole transcriptome, regardless of the number of samples you have on there. That's your assay costs. You will have sequencing costs after that as well. We don't get involved with that, so because it's usually so sorry. This is whole transcriptome for Geomex. I was talking about that because we were talking about comparing Visium to Geomex. Yeah. For CosMx 1,000 probes, the slide cost is $3,300 for the 1,000-plex RNA assay with no downstream sequencing costs. It's all done there on that.
That's about a third of the pricing.
Is it? Okay, that's great.
And you mentioned for the Geomex,
Yeah, they're just 27 base pair barcodes. And then in the collection plate, adapter sequences and everything, library prep to sequence for Geomex is essentially one PCR reaction. It's not like native RNA.
pretty older now.
Yes. You would get it in March if you placed an order now. That's where we are. We've pre-booked almost 70 of that. They start shipping in this year. Yeah.
Do you happen to know if there's any plan to expand this platform to other species besides mice and humans in the near future? Are allowed generally with rats and that sort of thing?
Yeah. It's funny. I had a call this morning with somebody at Duke who works with zebrafish. So CDC works with that. So we get this question a lot. Right now, the way to address that would be a custom assay. So still our bioinformatics team, you send us a gene list and they can build the probes and design all the probes according to what you need. With nCounter, we have a long history of working with all different species. So we already know, how to do that part of it. But it would be a custom assay build at this point. I'm hoping that comes, because like, it's not that, like, that's good for people doing rat work. So I am pushing hard for that, and the rest of my team goes internally.
So it's like one thing our labs sort of stuck with in one sense. It's like we would like to we're trying to do like a scaled down version of what this is from an off at the protein level. But we're kind of stuck playing the antibody game.
Yeah.
We're just trying to identify antibodies that can actually cross.
Yeah.
proteins of interest.
Right.
I guess for this is again going back to the idea of like crossover between species, and I talked with Nicole about this
Okay.
already. But like these validated antibody panels, I know that they're validated for the species that they're to target. Is there any information out there about like the validation as far as not picking up non-specific signal from like species to species? I don't imagine it's been factored in.
Yeah, so it wouldn't have been anything that we've tested, but I will say that, aside from the couple of morphology marker kits we have available off the shelf, we're not an antibody company. So we use commercially available antibodies. We have a morphology marker list that we maintain for projects we do in-house, and we tell you exactly the vendor, the solution. The validation for cross species would be done by the antibody vendor. Yeah. But there's something particular you're looking for, though. Let me know, and I can ask around. Okay.
Already see like if there's information about like which company they've seen.
Yeah, if it's the morphology marker list, it's listed right there.
Okay.
It's AppCam or self-signaling or whatever.
I feel like for a lot of those common markers, there's most likely
Probably.
nice friends.
And for the RNA side of things, we have a cross hybridization list with that. Do you have that
I'm
already?
like, I have that already. I'm talking about that. It's like, I feel like antibody cross-reactivity, it just seems more normal with somebody's done this.
Yeah.
I feel like probe specificity is a little tighter.
Yeah, I mean, it's predictive,
Yeah.
until we have an actual assay built. Yeah. Unfortunately.
So I guess this question, if you have a knock-in for human gene mouse.
Yep.
Could you use two different types of antibodies, like species-wise, or would you have to keep it separate from?
Just for an antibody assay?
Yeah.
No, you should be able to, thinking this through on the fly. No, you should be able to, if the product of your knock-in is truly human specific, and you're not going to get binding with any endogenous mouse. Might be close to it.
Okay,
Yeah.
so would you suggest we do mouse neuroscience or?
It's a mouse that you've knocked in a human gene.
We do like a mouse with a special antibody?
Well, what's the question? Is the question where this human protein is? Is the question how expressing it affects the environment it's in?
Yes.
Okay, then I would do a mouse assay and add in an antibody for it.
Okay.
And we can talk, more about that. Mhm. It's a great question. Humanized mouse models, we see those all the time for WTA especially, so totally doable. Just a little bit more thought has to go into your study design, but it's totally doable. Awesome. Well, thank you all so much. I really appreciate it. I think there's probably a lot of extra food, so help yourself, take stuff back to your labs.
Thank you to everyone who was able